Okay, you're really getting off the hook here because I think this slide I hope is review. So being that I'm not with you today, I can't put you in the hot seat. So I guess I'll answer these and I'll pause a little bit and see if you can kind of figure them out as I go through them. But basically, trying to answer these questions, if you think about what determines the primary structure, remember that's going to be, the inherited instructions, and so that's going to be DNA. And as far as what determines that final 3D shape, when you hear that final 3D shape, you should be thinking the tertiary structure. And that tertiary structure, remember all that folding and contorting that we did when we made our proteins at the beginning of the semester. The folding is due to the interactions between the R groups or the side chains that make up each individual amino acid. Okay. And think about when we built those proteins in class, I let you fold them however you wanted to, but I said if this were in reality, there would be something peeking over your shoulder and directing that folding. Much like, someone peeks over the shoulder of kids at a dance. And so we have chaperonins, or chaperone proteins, you can call them either way, okay, that basically direct and coordinate that folding. And then remember, back to our cell unit there, we can find ribosomes really in two places. The ones that are just loosely floating in the cytoplasm are called free ribosomes. And then we also have ribosomes that are attached to either some kind of surface, attached to the nuclear membrane, attached to the ER, to the rough ER, and so we call those bound ribosomes. And specifically in that cell unit, we talked about, well, what determines which ribosomes they're made at? And it really depends on where those final proteins are going to end up. So if they're going to stay in the cytoplasm, then it makes sense they're going to be made at those free ribosomes. If they're going to become an enzyme that's wrapped in a membrane, for example, the lysosome, they need to be made at a bound ribosome so that they can be packaged into a vesicle by the Golgi. And same thing here. So if they're going to be exported from the cell, so for example, hormones that will export and travel throughout the body, those will also be made at a bound ribosome. So I'm going to think optimistically that you all can still remember those things from earlier in the semester. This is kind of an interesting, really economical thing that cells do. In order to make protein in bulk, maybe they're cranking out lots of, let's say, insulin or something like that. Instead of using one messenger RNA and one ribosome, really here's the messenger RNA here. And we can have multiple ribosomes that latch on and read that code and make lots and lots and lots of polypeptides from that. So we call those polyribosomes. So really that's how the cells make protein in bulk. So it's really, really quite smart and very economical of cells to do that. Okay, so we're going to basically practice. This is what you'll need scratch paper for. So, and it's going to be paper that you give to me, but it can be, have something else on the back side of it. You'll get it back. But this would be something that I could ask you lots of different questions about. As you can see here, I've got 4 questions. But let's say I give you some kind of a template strand. Okay, so if this is the 3' end, and the first question is, I ask you, what is the complementary strand of DNA? Okay, so thinking back to Chargaff and his rules, okay, across from a 3' is going to be a 5', and then you just go through, and if it's T, A is going to go across from that. If it's A, you're going to have T, and if it's C, it's going to be G, okay, and so on. Okay, so that's going to be question number 1. Question number 2 then says what messenger RNA would be produced. Okay, And so again, if this is the template, okay, the messenger RNA, again, would start in the 5' to 3' direction, so it'd be 5'. If it sees a T, it's going to put an A. Okay. If it sees an A, that's when we use different code here, so it's going to be U. Instead of C, then it's going to be G. Okay. And so again, continue through the end of the strand. Okay. For what tRNA anticodons, okay, so we're talking, let me pick a different color here. If we were talking tRNA anticodons, okay, for this question, then you're going to look at the messenger RNA you just made up here, and in this case, it would be 3'. So again, A pairs with U, this is RNA. U pairs with A, and G, again, it wouldn't have to be perfectly complementary, but go ahead and make that UAC, and so on. Okay, so for every group of 3 codons here, you're going to have an anticodon that's complementary to it. Okay, and then finally, this fourth question, it says what amino acids would make up the final polypeptide. So this is where you're going to have the genetic code. I will always give you that chart. You're not going to have to ever memorize that genetic code. That would be silly. So for the amino acids, you need to look at your genetic code back at the beginning of the PowerPoint, or there's a copy of it in your book, or lots and lots of places online. But again, when you are looking at the amino acids, you have to look back at the messenger RNA. Okay, so for example, in the chart, if you find AUG, okay, that is going to code. Again, this would be the 5' end. It would code for methionine. So I'm just going to put MET. The 3-letter abbreviation is fine. Okay, and then again, for every codon you have here, it's going to code for 1 amino acid. Okay, so go ahead and use what you have written for number 2. Excuse me, to fill in the amino acids. Okay, so go ahead and complete all 4 of these steps for the entire strand of DNA, and then plan to turn that in to me when you walk in the door on Monday. And hopefully, if we don't have any mutation, everybody will have produced the same final polypeptide here. Okay. So the last part of this is really fascinating as far as the mutations that can happen. And so point mutations are very small scale mutations that can take place. Okay, small scale mutations, much different than the chromosomal rearrangements, the duplications and deletions that we saw in chapter 15 that can rearrange large parts of a whole chromosome. So when we're talking about small scale mutations here, we're talking about really one to a few DNA nucleotides are altered. Okay, and so the first of those, if we just kind of look at the pictures here, are substitutions. And so what I always do, just to help me with my reading frame, is knowing that the DNA is going to be read in groups of 3. The easiest thing to do is to draw a line, every 3 letters. That way it helps your eyes read it a little bit better and not accidentally skip a letter and make a mutation of your own. Okay, so again, notice for every group of 3 letters in the DNA, so for example, if we have ATG in the DNA, of course the shortcut is just change that T to U, and then you have the messenger RNA, AUG, which codes for MET. If we look at AAG, it codes for lyse or lysine. TTT, which will become UUU, codes for phenylalanine. That's the one that individuals with PKU cannot ingest. GGC codes for glycine. Okay, and so imagine up here that really this is the normal um, polypeptide. Okay, so this is pre-mutation. So let's say we've had a substitution mutation. And so in this case here, this one happens kind of towards the end of it. But here, instead of CCG, it's mutated to CCA. Okay, again, maybe this mutation happens when DNA is replicating. Maybe we have some kind of radiation exposure or something in our food or stress or smoking or tanning beds, you name it, where now CCG is mutated to CCA. Okay, and so then the other strand of DNA is going to be GGT. And so when the RNA is transcribed, okay, it's GGU. So if you take a look at the chart, let me flip to my chart here, okay, our final thing here, this is still going to code for MET, okay. This one is still going to code for lysine. This one, phenylalanine. And then this one, if you take a look at the chart, it's one of those redundancies. So regardless if it's GGC, GGU, it's still going to actually code for glycine. Okay, and so this mutation happens to be called a silent mutation. Okay, because it still codes for the exact same amino acid. Okay, so it's a silent mutation. Okay. This one here is called a missense, okay? And so again, if you take a look here, in this case we have still a single point mutation. It's a substitution of one letter, but that one letter happens to be, instead of the third letter in the codon, it's actually the first, okay? And so instead of having GGC, it's going to be AGC. Now that's probably going to give us a different amino acid. So if you take a look at your chart, okay, again, AUG is still going to code for methionine. Okay, this is still going to be lysine, phenylalanine. But then when we get to AGC and you look for that in the chart, you're going to find it does not code for glycine anymore, it codes for serine. Okay, we have a different amino acid again. So that could be significant. Okay, that's a missense. This is an example of a significant one. So here we have a single amino acid that's different. And so in all of our bodies, one of the amino acids that's essential to normal hemoglobin is glutamine. But in someone who has sickle cell, notice they have just a single point mutation. So instead of CTC, it's CAC, so that second letter in the codon is different. So instead of GAG, we're looking for GUG in the chart, and now it codes for a different amino acid. Okay, because when that hemoglobin folds, it still doesn't end up with quite the right shape, and so this person would have those sickle-shaped red blood cells, which are not very good at transporting oxygen. These sharp little edges of those red blood cells can get lodged in the capillaries and really cause extreme pain for individuals with sickle cell anemia. So here's an example where just you think one letter in the DNA is not going to be significant, but it really is. Okay. So what I would like you to do, we've got a couple slides here left. If you take a look, this is a nonsense mutation. So what I would like you to do is figure out what amino acids are going to be coded for. Okay, so basically use this strand of messenger RNA and compare it to your chart. You know, look up AUG and see what that codes for. You probably know that already without looking that up. Look at UAG and see what that codes for, and so on. Okay, so you can put that on the bottom of that other page that you did. Okay. Same thing for this one. This is a deletion mutation. So instead of a substitution, it's a deletion. And it's going to lead to a frame shift, and you'll see why. So again, comparing to the normal that's up here on the top, go ahead and read this strand right here. and fill that in.